Hello friends, warm welcome in this video, that is the urea cycle. Friends, the amino group of amino acids are used in the biosynthesis of various other amino acids, nucleotides and biological amines. If these amino acids are not used in the synthesis of all these compounds, then the amino group will be channeled into a single excretory end product. The animals may have various modes of excretion, that is amniotic, ureotelic and uricotelic. Most of the aquatic species such as the bony fishes are amniotic. That means they are excreting amino nitrogen as ammonia. In this organism, the toxic ammonia is simply diluted in the surrounding water. Terrestrial animals require pathway for nitrogen excretion that minimize toxicity and water loss. Most terrestrial animals are ureotelic, means that they excrete amino nitrogen in the form of urea. On the contrary, birds and reptiles are uricotelic, means that they excrete amino nitrogen as uric acid. But plants recycle virtually all amino groups and nitrogen excretion occurs only under very unusual circumstances. In uricotelic organism, the ammonia deposited in the mitochondria of hepatocyte is converted to urea in the urea cycle. This pathway was first discovered in 1932 by Hans Krebs and a medical student associate Kurt Hanslieb. Sir Hans Adolf Krebs discovered this pathway 5 years before the discovery of TCA cycle or Krebs cycle. Urea production occurs almost exclusively in the liver and in some extent in kidney, and thus is the fate of most of the ammonia channeled here. The urea passes into the bloodstream and thus to the kidney and is excreted into the urine. The urea cycle begins inside liver mitochondria, but 3 of subsequent steps takes place in the cytosol. The cycle thus spans 2 cellular compartments, that is mitochondria and the cytosol. The first amino group to enter the urea cycle is derived from ammonia in mitochondrial matrix. The liver also receives some ammonia via the portal vein from intestine from the bacterial oxidation of amino acids. Whatever its source, the ammonia generated in liver mitochondria is immediately used together with CO2 in the form of bicarbonate produced by mitochondrial respiration to form carbonyl phosphate in the matrix. This ATP dependent reaction is catalyzed by carbonyl phosphate synthetase first. This carbonyl phosphate synthetase first is a regulatory enzyme. This mitochondrial form of the enzyme is distinct from the cytosolic form, that is carbonyl phosphate synthetase second, which has separate function in pyrimidine biosynthesis. The carbonyl phosphate, which functions as an activated carbonyl group donor, now enters the urea cycle. This urea cycle has 4 enzymatic steps. In the first step, carbonyl phosphate donates its carbonyl group to ornithine to form citrulline with the release of inorganic phosphate or orthophosphate. Ornithine plays a role resembling that of oxaloacetate in the citric acid cycle, accepting material at each turn of the cycle. This reaction is catalyzed by ornithine transcarbamylase, and the citrulline produced in this step passes from the mitochondria to the cytosol. In the second step, ammonia group now enters from aspartate that is generated in mitochondria by transamination and transported into the cytosol. Amino group now enters from aspartate by condensation reaction between the amino group of aspartate and the urido, that is carbonyl group of citrulline, forming arginosuccinate. This cytosolic reaction, catalyzed by arginosuccinate synthetase, requires ATP and proceeds through the citrulyl AMP intermediate. Now this arginosuccinate is cleaved by arginosuccinase to form free arginine and fumarate. The fumarate so formed enters into the mitochondria to join the pool of citric acid cycle intermediates. This is the only reversible step in the urea cycle. In the fourth and last reaction of the urea cycle, the cytosolic enzyme arginase cleaves arginine to yield urea and ornithine. Ornithine is transported into the mitochondria to initiate another round of urea cycle. Friends, the enzymes of many metabolic pathways are clustered with the product of one enzyme reaction being channeled directly to the next enzyme in the pathway. In the urea cycle, the mitochondrial and cytosolic enzyme seem to be clustered in this way. The citrulline transported out of the mitochondria is not diluted into the general pool of metabolites in the cytosol, but pass directly to the active site of arginosuccinate synthase. This channeling between enzymes continues for arginosuccinate, arginine and ornithine. Only urea is released into the general cytosolic pool of the metabolites. Friends, the citric acid cycle and urea cycles can be linked because the fumarate produced in the arginosuccinate reaction is also an intermediate of the citric acid cycle. The cycles are in principle interconnected in process which is dubbed as Krebs bicycle. However, each cycle can operate independently and communication between them depends on transport of key intermediate between the mitochondria and cytosol. Several enzymes of the citric acid cycle including fumarase, that is fumarate hydratase, and malate dehydrogenase are also present as isozymes in the cytosol. The fumarate generated in cytosolic arginine synthesis can therefore be converted to malate in the cytosol, and these intermediates can be further metabolized in the cytosol or transported into mitochondria for use in the citric acid cycle. Aspartate formed in mitochondria by transamination between oxaloacetate and glutamate can be transported to the cytosol, where it serves as nitrogen donor in urea cycle reaction catalyzed by arginosuccinate synthetase. These reactions making up the aspartate arginosuccinate scent provide metabolic links between the separate pathways by which the amino groups and carbon skeletons of amino acids are processed. So friends, if you like this video, please subscribe to our channel, press the bell icon and share this video among your friends. Thank you.